Dr. Neetu Singh, Center for Biomedical Engineering, IIT Delhi. Dr. Singh obtained her bachelor's and master's degrees from University of Mumbai, after which she moved to USA to perform doctoral work in chemistry with Professor Andrew Leon at Georgia Institute of Technology, Atlanta. At Georgia Tech, she worked on developing novel synthetic routes towards the design of multifunctional hydrogen nanoparticles. Following her PhD, she moved to Cambridge to pursue her postdoctoral studies at the Harvard MIT Division of Health Sciences and Technology with Professor Sangeeta Bhatia. In the Bhatia laboratory, her research efforts were focused on developing nanomaterials for achieving and investigating RNAi therapy. Dr. Singh returned to India and joined as faculty in the Polymer Sciences and Engineering Division at the National Chemical Laboratory, Pune, 2012. Her work is published in very high impact journals and also gathered a lot of attention and featured in faculty of 1000 Prime, identifies top research in biology and medicine and several other science media outlets. Her important scientific contribution in developing strategies for managing cancer got her the Kiran Shaw Muzumdar International Oncology Fellowship for establishing cancer-based research ties with MIT USA. She is also a recipient of the Innovative Young Biotechnologist Award 2013 by DBT India. Dr. Singh joined as an assistant professor in the Center for Biomedical Engineering, IIT Delhi, in April 2014. In her current program at IIT Delhi, she is establishing a research program that explores systematic probing into nanomaterials, biological activity, and formulates design rules for developing biosystem for specific biomedical and technological applications. Her area of interests include structure activity relationships in design of biologically relevant nanostructures, small functional nanomaterials, drug delivery, biosensors, biomedical implant coatings, tissue engineering. We welcome you, ma'am. Over to you, ma'am.
Thank you so much for this introduction, Sai. Is that correct? Yeah. So, it's a thank you so much, Dr. Jairam, for giving me this invitation. I think it's an honor. The reason is because I myself, I grew up in Mumbai, and, I'm a product of Mumbai University. And the training I got in Mumbai University is, I've been to many universities here in India, and also I've traveled, I've looked at their curriculum and everything. I think it's different. I mean, we get trained in a different way. So I'm thankful to my university and alma mater, for giving me the training. And you all are at a very good place. SP College, of course, we all know it's such a wonderful engineering college. So I'm very glad that I'm presenting my work here. So thank you so much for giving me this invitation. So I'll share my screen, just a sec.
Yes, ma'am.
Yeah, can you all see the screen now?
Yes, it is in process. Yeah, it is
Okay.
visible.
So, what I'll do, I will try to do it in presentation mode, but many a times, during this online presentations, the screen does not move with the same rate. In case that happens, please let me know that the screen is not moving. Okay.
Yes, madam. Now it's not in presentation mode still.
Oh, so see, that is the reason. So what I'll do is, I had it in presentation mode because there is a lag, so it usually does that. So I will not do it in presentation then. Let me try one more time. Is it in presentation mode now?
No, not it. Madam, maybe you have to share entire screen. There are two options. One is entire screen and another is
So it doesn't work, because there is basically sometimes, let me do stop sharing.
entire screen and the another is the window. So if you
Window,
choose
yeah.
window, then this problem generally happens.
Okay. So I'll do entire screen. Yeah, can you see the screen now?
Yes, it is in transition.
And
Yes,
this
now it's perfect. Yeah, it's
okay,
perfect.
great. Thank you. I didn't know this. See, we're learning still. Anyway, so the topic of my talk today is engineering new materials for drug delivery. And I call it a chemist perspective because, as I said, I am, in introduction, you learned that I'm a chemist by training. So I got my PhD from chemistry department and then moved to do biomedical research during my postdoc. and of course during the later stages of my PhD. So so the whole talk is basically geared around this concept of drug delivery and what are the challenges in drug delivery. And what I'll try to do in my talk is explain to you that how a chemist can look at these problems in a different way, and how we can give solutions from a chemist perspective. There could be many solutions to the same problem. However, the solutions that my lab is working on has a lot of chemistry impact on it. Okay. So before I get into it, I would like to tell you what are the things that we're doing in our lab at IIT Delhi. And I invite you all to, whenever you're in Delhi, just shoot me an email that you would like to visit our lab, and we'll show you all the exciting things that we're doing. So the lab that we've set up and the problems that we're taking on are very simple. Basically, I'll give you one quick example. Tumor biopsies are very common procedures now, and it's very easy to get a biopsy. I mean, it is one of the things that happens when someone is to be diagnosed with cancer, right? So tumor biopsies is very easy to obtain. Most used clinical practice even today, right? From this tumor biopsy, it is very easy to obtain the tumor cells. So going from A to B is a very simple process. You can get all these, you can homogenize it, you can extract the cells. You can also extract specific cells that you're interested in. The question we asked is, can we go from B to A? Why is that important? Because if we can go from B to A, I can actually create, so I got only one biopsy, which is really small. From this, I can create multiple tumors. If I can go from B to A, I can assemble multiple tumors from the patient. So it could be patient specific. And I can use these tumors to do drug studies. Just giving you a simple example. I can look at which drug combination actually helps in getting the tumor size reduced, right? Or killing the cells. So the idea was, can we go from B to A, create in vitro models, and can we utilize these cells that are coming from the patient or our primary cells to understand and intervene in cancer, right? So that was one problem, and my lab is putting in a lot of effort in doing that. It turns out going from A to B is quite simple. However, from B to A is not that simple, okay? The next question we asked is, so this is basically, the first thing that I talked about is basically the biosensors work that we're doing. And the other one is basically, tissue engineering. So here what we're doing is, if you look at this structure, this is your bone and this is your cartilage. And, you will see that everywhere, the way nature, our body has it is that there's an interface, there is this, and we know that the materials property of your bone and the materials property of the cartilage is very different. Cartilage is slightly softer than bone. Of course, it is not as soft as the tissue, but you also have regions which are soft tissue. So in the same, like for example, near the bone, you will have three different materials property. You will have a very, very hard bony structure, which is also brittle. You will have a cartilage which is very strong. You can, compression is very high, compression strength is very high. And you will have muscles or other tissues, softer tissues, where elongation is higher and the property is totally different. The question is, how does nature do this? How does nature control that the specific architecture that I'm talking about, that only this region will have cartilage, only this region will have bone? Or, if you look at other organs, for example, a liver, it has even more interesting structure. It has got a hexagon structure, and in that hexagon, you have beautiful liver cells arranged. Just Google in your free time, Google liver architecture, and you will find that it's a beautiful hexagon structure. How does nature obtain this, organized geometries? So we thought that, that now this is a challenge, because till today, you're not able to get cartilage and your bone on the same substrate without, simply without giving any cues or without So the way it is done is you have two different materials and you stick them together. You don't grow them, let them grow in one structure. So the question is, how can we mimic the alignment of these multiple cells and obtain the function that organs have? So we are spending a lot of time, and we have published quite a lot of work in this area. And of course, it's an ongoing journey. We have not, of course, completed. I mean, we're still learning a lot. So the third area that we work on is the drug delivery, and I'll be talking about drug delivery a little bit more detailed in today's talk. And that is, so in this one, basically we are trying to look at the challenges that are there in drug delivery. For example, delivering of hydrophobic drugs is a challenge. So, liposomes, you all are aware, even with the vaccine, I think it has become even more common. So liposomes are very commonly used drug delivery vehicles, and we are looking at alternatives to liposomes. Okay, so that brings me to the introduction of my concepts of my talk. Basically, you all have heard of nanomedicine, and you all know how nano is important in drug delivery, right? Because it can actually give you more surface area, and it can allow you to do more surface functionalization, load drugs with higher concentrations and things like that. So a typical approach for developing a nano drug delivery carrier is called the bottom up approach, where what you do is you design a system and you use it for various applications. So here, you have liposomes I was just talking about. You can have quantum dots, you can have gold nanoshells, you can have mesoporous particles. There could be carbon nanotubes, dendrimers, magnetic particles, micelles. All of this has been explored quite extensively. Okay, of course, you try and make it. You can quote polyethylene glycol, PEG. We'll talk about PEG again in the next few slides in more detail. So polyethylene glycol will give the stealth property, or stealth is basically the immune system will not recognize the nanoparticle. Okay. So the idea is you have these various materials, you form, particles in the size regime of less than 100 nanometers, and that you use for delivering of cargoes. The cargo can be nucleic cargoes, the cargo can be small molecule drugs, it can be anything, right? So with this aim, because you remember we have to use this to deliver, you require this vehicle to go. You want to put an address on the vehicle, right? You want this to go to a particular region. So, for example, if there is a lung cancer, and you inject it, it should not go to every organ. It should just reach the lung, because that is where it is required. You cannot inject it in the lung because it's an internal organ. So delivery at the position of internal organs is slightly challenging. So the way you do it is you give an IV injection to the patient, and the drug basically enters and goes there, right? So this is basically to achieve that. What people have done is they have used these something called as multifunctional nanoparticles. So they have nanoparticles where they will put in targeting agents. They will also want to look at, we become a little ambitious, and we want everything from one thing. So there will be therapy, there will be diagnostics. I can do imaging of these particles, I can identify where they are going, so all these parameters are actually put into these particles. Okay, so unfortunately, even with so much of work, translation of these things in clinics is very limited. There are very few nanoparticles or multifunctional nanoparticles which are there in the clinic. So what is the problem? The problem is, every time these nanoparticles are taken up into the stages like clinical phase trial, phase 1 or phase 2 or phase 3, they fail. Okay, because they fail, they are not able to get into the clinic. Why is that? And we will learn, because so clearly the bottom up approach is not working. So there is a need for a new approach to address this problem. And the new approach we propose is a top down approach. What is a top down? Top down means you have to first look at, like, piece out this whole thing. Okay, so you understand how is this, what kind of an interaction do you require from the nanoparticle, what kind of a uh, for example, you have the nanoparticle, it should go and target the lungs. What kind of cells do you have on the lungs? What kind of receptors can you utilize for lungs, targeting the lung and things like that. You do this fundamental understanding. With that fundamental understanding, you come up with this process where you design, you understand this extracellular interaction and the intracellular interaction. You then design your material and characterization, do some iterations, and pursue this cycle. Finally, you end up with something which is giving you some promising results, and that can be explored for new drug delivery applications. This will allow you to, so earlier approach, the bottom up approach, there you're making new particles, you're creating new entities. Okay, so every time you create a new particle, there is approval that is required, and you take it to a certain level, and then you remove, they're not successful, so then you have to give up that process. However, in the top down approach that I just discussed, you can have the material, the same material which did not work, you do an understanding, do the iteration, change the material a little bit, and then do these applications. So the amount of time it takes to get approvals for the material is reduced. Because of that, you fast track getting clinical approvals, and you might get things in the clinic. So this is the basic difference between the two approaches. Well, what kind of things can you do with a nanoparticle? So for example, when I talked about iterations, what kind of iterations can you do? Well, you can change the shape of the material. For example, you can make it into a rod shape, a sphere, an ellipse, a plate, star, cube, whatever you want to call it. You can change the structure of the material. You can have a core shell structure. You can put like an antibody kind of a thing. You can have a carbon, these long tube-like structure, where you can fill in things. You can have, again, depends on what material you're using, whether you're using liposomes, you're using metals, you're using proteins, depends on, or any sugar, depends on what it is. Okay. You change the size. For example, you can go from 1 nanometer. I told you that you have to be below 100, right? So you change it like really small, less than 10 nanometers, about less than 40 nanometers, less than 80 nanometers, and things like that. You can change the surface characteristics. For example, do not have groups which will allow hydrogen bonding, change the charge on the surface, change the surface groups, put targeting groups like antibodies and carbohydrates and other things. You can make them biodegradable and biocompatible. So anyways, so various strategies exist, right? Now, looking at what results, as I said, if this information is there, then why can't you just utilize this information and make a better material, right? Why is still the nanoparticles very toxic? Well, so let's spend some time understanding what is the reason for the toxicity of nanoparticles. So for example, it is now understood that if you have a small size, so for example, you have a really small particle, less than 10 nanometers, there are a lot of proteins which are less than 10 nanometers, right? What will happen to and these nanoparticles are going to interact with those proteins. So if you have something of similar size, what will happen? Will you have sterics there? Yes, right? And we'll come to this concept of sterics later on in my talk when we talk about, is there a chat happening also? Okay. All right. So I'm not able to see the chat on this window because everything is in presentation mode. So I'll respond to those things later. Okay. So there is basically, if it is a small size particle, very small, it is in the same size regime as the protein. So because of the interaction, there will be differences in protein folding. The protein folding can get interrupted. The proteins can get denatured and things like that. Okay. So anything that small is actually not good. However, once you make them bigger, the cellular uptake decreases, and then what happens is they end up getting less inside the cell and getting at other places. So then what is the ideal size? It is understood that about 20 to 30 nanometers is something which is good for uptake, but then the toxicity will not be that much. You can, if you have a shape argument, spherical nanoparticles are basically, they have the highest uptake. However, they also have very high toxicity because they can easily, interact with the cell membrane. Whereas if you have like a rod, for example, this, depending on what the aspect ratio of this rod is, so it's an anisotropic structure size, right? So depending on the aspect ratio, let's say it is, few nanometers thin, like in this dimension, and few like hundreds of nanometers long, right? um, Larger particles will just sit there, so the toxicity is reduced. However, they cannot enter inside because they're too large, and that is why the uptake is diminished. There was a very recent science paper, like not a recent one, but about a decade ago, which talked about that they actually get inserted inside like a needle. So if you have the thickness really less, like few nanometers, then it becomes almost like a needle, and it can pierce through cell membrane, which actually can cause toxicity. So again, it's not very clear, but what I'm trying to tell you here is that shape of the material can also cause toxicity, even though the composition is exactly the same. Talking about composition, like metal particles are quite commonly used in drug delivery. Now, if the metal has some impurity or it is doped with something, it can actually degrade and cause a lot of toxic effects. So, for example, quantum dots or inorganic quantum dots made up of cadmium and tellurium and things like that, they're very toxic, okay? Not because the nanoparticle is toxic, but the elements that are used there are quite toxic to the cells. So, how pure your material is also important. Otherwise, you'll get a lot of leachant. Same thing with a polymer. If you have a polymeric material, if there is any unreacted monomer left, that can cause toxicity. Okay, and a typical example is example of bone cement, where you have methacrylates, and these methyl methacrylates, and these methacrylates are actually, a lot of monomer can still remain unpolymerized. Okay. Um, you can also have metals which are leaching out. So, for example, coordinated, and then it stays in solution, and then it slowly leaches out due to acidity, it leaches out, or any reason. So, that is very important, and that can cause toxicity. Um, then, nanoparticle stability is also important for the toxicity. For example, um, suppose something is degrading, and the degradable material or the degraded product is toxic, uh, and this degradation gets enhanced in presence of uh, low pH or high pH. For example, acid catalyzed hydrolysis. Okay, so if there is inflammation, the pH is really low, and if this material goes in the region of inflammation, the hydrolysis will start happening really fast, and because of that, you can have products, byproducts as a result of hydrolysis, which are coming faster, you know as the anticipated compared to what was anticipated, and that will cause toxicity. So stability of nanoparticle is also extremely important. The surface charge on the nanoparticle is also important, because you can have, for example, if it's a highly positively charged particle, the surface of the cells are negatively charged, and if it is highly positively charged, it can interact with the negative charge with electrostatic interaction, and because of that, it will just rupture the cell membrane. Okay, and that will be very toxic. So as I said, there could be many reasons for toxicity. uh, these are, about the nanoparticle, something that happens in that is basically the the physicochemical property of the nanoparticle, right? Other thing which is important to understand is that in the host or in the humans, um, reactive oxygen species are quite commonly generated. So these reactive oxygen species are actually species which are ROS, we call them, in short, are one way of getting rid or denaturing proteins which are not important. Okay, so getting rid of the debris or getting rid of unwanted proteins, ROS is used to degrade the products. Now, nanoparticle surface actually can act catalytically and increase the ROS production. Because of the increased ROS production or catalysis of ROS, what will happen is there is an oxidative stress in the system, and it can actually cause DNA damage and things like that. And this ROS becomes very, very dangerous. So, this is also a reason for toxicity. And if you want to mitigate it, if you want to make it, so as I said, if you want to mitigate the toxicity, you can either change all these properties. However, I'm sure it is clear from the physicochemical properties that changing the physicochemical properties too much, for example, I change the shape, but I'm going to compromise on the other properties, right? So that will actually be not very good, and that will not be a solution. So how do we mitigate toxicity? And that is why I started that is why I'm trying to tell you here is now you have to understand what happens if the ROS is generated, what happens, and how do I stop the ROS production. Remember, the ROS is not being produced by the nanoparticle. The ROS is already present in the system, right? So to mitigate, we'll have to come up with strategies to catalytically activate the ROS production. Again, keep these thoughts with you. We will utilize this in the later section of the talk. Um, then the second part is once the nanoparticle enters, what is the pharmacokinetics like? What is the absorption profile? Where does it get distributed? How is it metabolized? Where is it excreted? If we know all these applications, we will be able to modify the material in such a way that you get your desired result. For example, maybe it's very toxic because if it aggregates. But even before it aggregates, if I know the pharmacokinetics, I let it reach the site. Once it reaches the site, the majority is at the organ site, and it will have less systemic toxicity, right? So these are the concepts that you could utilize. All right. See, the other thing I could do to mitigate cytotoxicity is avoid protein corona formation. And I'll talk about protein corona formation in a little bit. Change the intracellular interaction, decrease the ROS generation. So this is basically three things that you could do to do that. And my talk today is going to tell you how we have put in efforts in each of these three categories to create solutions which reduces the toxicity associated with nanoparticles. Okay, so let's try to see. I mean, let's talk about the protein corona formation, and let's try to now understand, get a little deeper, and try to understand what happens if you have the nanoparticle. It goes in this system. The first thing it will see is the proteins, because it gets injected into the blood stream, and the bloodstream will have your plasma proteins, and those proteins will start to interact. So I'm not the first one talking about it. There was a beautiful paper published in 2009 in Nanoletters and 2014 in Biomaterials, which talked about various aspects of the nanoparticle, various physicochemical properties of the nanoparticle, and how that affects the protein interaction so if the protein interaction gets affected, the cellular interaction will get affected. So for example, size, what happens, there is an optimum size. This is 40 nanometers, for it to work better. Then cellular uptake, what happens to cellular uptake? There's an optimum size. What does the shape, depending on the aspect ratio, there is a bell curve, like there is a optimum shape or the aspect ratio that you need to maintain, the mechanical properties, surface charge. A lot of work has been done and can be utilized. So here is what happens to the nanoparticle. You have proteins in the solution and they will interact with it. If the protein on the solution in the plasma interacts with the nanoparticle, what will happen is, remember the specific functionalization that we did in earlier slides where we made it multifunctional? If the protein comes and sticks there, we will lose that. So all the specific ligand will be masked. The charge, remember I was playing up with the charge here to change the interaction. What will happen to the charge? They will get masked. Functional groups will get masked. And because of masking, you will get unwanted immune response. So these are all negatives with the protein corona formation. What are the advantages? Well, because if you have the protein on the surface now, which is from the host, this is not going to get rejected. It will end up, in circulation for a longer time. So it depends on how, we utilize this protein corona formation to create systems which are better drug delivery vehicles. So, again, I don't have to talk about why use nanoparticles for drug delivery, but now that I'm going to tell you, so basically the lesson that I want you to learn from this is that protein corona can be advantageous as well as disadvantageous. Most of the time, it is a disadvantage because unless I know what specific protein interactions are, I will not be able to develop a good solution. So either I know which proteins come and stick there, or I consider this as a disadvantage and come up with strategies to reduce protein adsorption. Okay. Anyways, so why use nanoparticles for drug delivery? Well, because we require, a lot of nanoparticles are insoluble. A lot of drugs are insoluble in water, and we require some vehicle to deliver these insoluble drugs into the host, which is, host is an aqueous medium. So because, what happens is if it is hydrophobic, it enters, they aggregate and gets cleared by the kidney. Okay, so you have to use effectively a very high dose. To avoid this, you can use nanoparticles, and liposomes are one of the successful models. And as I said, we'll discuss that. Why nanoparticles will be important? Because nanoparticles, so by the way, nothing can pass through our vasculature, So for example, blood, has passages by which are capillaries and other things, by which it enters the organ. It just doesn't get absorbed through the lining of our blood vessel because the endothelial cells are very tightly closed and in normal situations, and they don't let even oxygen pass through. Nothing passes through this tight network of cells. So this is the picture here. Whereas if there is some inflammation or cancer, actually the junction between the endothelial cells is broken. So you get some gap in between and things can easily pass through. That junction is not like in micron size, okay? It's in few hundred nanometers. What it means is that if you have a nanoparticle, it can selectively go through that. The vasculature is leaky, technically that's what we call it. So it can leak. And because it leaks, the nanoparticle can selectively enter that region. So this is called as the enhanced permeability and retention effect, or the EPR effect. And there is more tumor accumulation. This is part of passive targeting. Passive means you're not done anything to target. It just ends up there. You can also do active targeting, where you can put a functional group which binds to the particular receptor. So I'm sure you understand the importance of receptor with the pandemic. The ACE2 receptor we keep talking about is present in the lungs and the respiratory regions. And this is how things get selectively enter the lungs, okay? And this particular virus that we're talking about utilizes that. So same way, you can utilize the receptors on the cell surface to deliver your nanoparticles, okay? And that would be your active targeting. So this is a protein corona, what I was talking about. So you have a nanoparticle, you have different layers of protein, and again, there is also a process by which this whole protein corona formation happens. Okay, so you have a first layer of protein, second layer of protein, and all that. Um, so to avoid this protein corona formation, basically, scientists have done a lot of work, which is just make the nanoparticle hydrophilic, because if you make it hydrophilic, it won't get into. So here, you change the material, you change the surface, you change the size, you change the charge, you change the shape. Whereas in the environment, factors that will affect is the composition. For example, there are no proteins in the region that you want to send. The protein corona will not form. How long does it stay in that particular region? Exposure time to that region is important. What would be the pH of the region? That will become very important. Temperature and shear stress. Temperature is not that important because most of the time the differences are very low. Shear stress is important, whether it's in flow, it's in static place, it just goes and gets stuck somewhere or things like that. So that will become very, very important. So what to avoid that, people have started to utilize polyethylene glycol, okay? Or polyethylene glycol has resulted into something called as liposomes, and they're coated with polyethylene glycol. So typical liposome looks like this structure. You have a lipid bilayer, you have an aqueous core where you can entrap your drug, then you have a lipid bilayer, and on top of it you have your polyethylene glycol. By the way, this is in clinic. Doxil is a liposomal formulation of doxorubicin, a very important chemotherapeutic. So this is in clinical use. Then there is Abraxane. So instead of using a liposome, I could use the molecule, conjugate it with albumin. Now, albumin is the most abundant protein that is found in the plasma. So if you have albumin on the nanoparticle, then the system will not be able to recognize it. It will just think that it is a part of the whole system. So Abraxane utilized that concept, and it's a albumin-bound paclitaxel. Okay. So does that mean that liposomes are a complete solution? I mean, even with the vaccine, liposomes are being used, no? So do we have a complete solution? Unfortunately, no. Yes, liposomes are a biocompatible material. They can encapsulate hydrophilic as well as hydrophobic drugs. For example, the hydrophilic drugs can go inside the core. The hydrophobic drugs can partition here. So it is useful. You can put polyethylene glycol to make it more biocompatible, but use of liposomes have shown to induce anti-PEG antibodies in some individuals. There is a very common effect of complement activation related pseudoallergy, or the CARPA effect, which is hypersensitivity within minutes of injection. Okay. Now, again, remember our vaccines also have this polyethylene glycol, and you must have heard that they talked about that this is a very common allergy which is seen with PEG when they were talking about this one, the Moderna and the Pfizer vaccines. And they said that, there's a little bit of allergy with the component, which is polyethylene glycol, which is used in these vaccines. And you get this is because of this CARPA effect. Okay. So clearly, liposomes are not a solution. We are using them because we don't have anything better. So our lab said that can we provide a solution for that with the understanding that I've just talked about. So we started looking at how does nature does this, I mean, remember, whatever we are trying to do today in health is basically mimic nature, because nature has a healthy body. We have changed something in our healthy body. So to fix it, we'll have to look at how does nature make it perfect, right? So that's the whole concept. So we started looking at how does nature deliver things. So the first thing that came to our mind is RBCs. Now, red blood cells, again, I'm a chemist, so I don't understand the biology of RBCs. But what I looked at, when I looked at, what I understood was that it is one cell which is used to carry oxygen, okay, to different regions. How does it do that? It does it with some complex called as the heme. So carrying of oxygen is not important. But if you had just heme coordinated with your oxygen, probably it won't work. That is why nature has a carrier, which is your RBCs, right? Why did nature not use just hemoglobin protein that you have with oxygen and that could circulate? Why are we trying to do albumin with drugs similar to Abraxane and that there's no reason, right? If nature is using a carrier, then clearly it has a mechanism which would clear the protein if it is flowing freely. And that is what happens with our systems. So we thought, let's use this RBCs as a delivery vehicle. So we started looking at RBCs as a chemist and started looking at the structure of RBCs. Okay. Turns out that the RBC is a very nice biconcave structure. It's about 7.5 micron lengthwise and 2 micron thick. Okay. So it just looks like this kind of a structure. Okay. It has no nucleus, so you don't have to remove it. Literally a bag. It does not have a lot of organelles, but it contains the heme. So it is delivering the heme, which is not the main protein that it needs to take. It needs to take oxygen, which is binding to the heme. Okay. So, and by the way, the RBCs are cells which are in circulation. They don't get stuck anywhere. They're circulating. So we thought probably the concave structure allows them to, the fluid dynamics is changed because of the shape of RBCs. We don't care about fluid dynamics so much because this is 7.5 microns. So if you want a micron sized in a really tight, the capillaries are few millimeters, no? So that's a very narrow place for it to go. So then I have to play around with this structure or the shape to get a good flow. If I have nano, I don't care about the shape because I can have any shape. So the shape was not very important for us. So we thought, then once the shape is removed, then what is there in the RBC? Nothing. It is just a self-assembled lipids giving you the cell membrane with some proteins in them, right? Why should I? So we were very excited. We thought, can we use this RBC to deliver, whatever is required? I remember, and it can stay in the host for a long time. So what it means is that if we use these RBCs, they will be in circulation for a longer time. It will be able to reach the site by EPR easily, tumor site by EPR easily. It can also, can we use it to deliver hydrophobic drugs was the question. Then we started looking a little deeper into the RBCs and started thinking, why has someone done anything with it? And we found that we're not the first one to use this idea. Many people have utilized it. And what they've done is RBCs are stealth. They have stealth. The system, body does not recognize them as foreign, okay? And that is why it is in circulation. How does it do that? It has something called as CD47, or it's a clustering protein. So CD47 is on the surface of RBCs. We started looking at the biology of RBC now. This CD47, macrophages are cells which are there to remove anything which is not needed in circulation. So they are basically, they phagocytosed. Phago means bigger. Anything larger, smaller things can be taken up by any cells and cleared. However, for larger cells, so a cell typically is about 10 micron. So anything which is larger than 10 micron, you need a larger cell, and macrophages are big cells. Their scales are about 500 microns and things like that. So they can take up bacteria. They are in place to remove all the debris, bacteria, dead cells, and things like that. Okay. So if anything can clear RBCs, the only thing that can clear it is macrophages, because RBCs are also small, but in micron size, right? So the macrophages are the only ones that can clear. Macrophages has this something called as the signal regulatory protein alpha, or SIRP alpha. This SIRP alpha interacts with CD47. Because of the interaction, there is a signal which is given, which is called as clustering. The moment clustering happens, the signal will go to the cell that, oh, this is not foreign. This is the same host ingredient, and don't take it, don't digest it, okay? And that's how the RBCs are able to uh, get rid of the macrophages. They don't get trapped by macrophages. Now, uh, again, I'm not the first one looking at this understanding. People have looked at it. So they said, okay, great. Why can't we put CD47 on the nanoparticles? And then, macrophages will say that they are not self and they will not get cleared. First, if we put CD47, protein adsorption can happen and you can get masking of this. But let's say I avoid that. I put polyethylene glycol, because polyethylene glycol is very hydrophilic. It does not allow proteins to interact. So I have polyethylene glycol here. What will happen is this will interact. Because of that interaction, it should not get taken up. Now, remember, this is a clustering effect. So again, forget about the biology. As a chemist, what I see is that the density of CD47 becomes very important, because if I have one CD47 on, are you all able to see my cursor also? Yeah?
Yes, ma'am.
Okay. So if you have CD47 on one CD47 on this side, another CD47 on this side, will you see clustering? No, right? You will not see clustering. Will that strategy work then? No. I don't know what is the distribution of CD47 on RBCs. That is impossible to identify. So I won't be able to create a strategy which is putting CD47, and that is exactly what happened in the literature. It failed. Okay, forget it. I don't want to do CD47. What I'll do is I will take the RBCs and coat it on the nanoparticle. Well, that's what many people have done. Unfortunately, it turns out that if you do that, this is micron. You will have multiple nanoparticles encapsulated in it. And again, you will be making carrier for the nanoparticle, not the nanoparticle will not become the carrier. Okay, which is not a good idea, because then you won't be able to release the drugs and things like that. So that also did not work. A lot of people have put RBC surfaces on the nanoparticle. A lot of work has been done on, again, making them nano also, taking part of the surface and putting it on the nanoparticle. Not successful, because remember, the orientation of CD47 might change. Okay, so a lot of things can happen. So then, what is the alternative strategy? The alternative strategy, we thought, is you have the erythrosomes, erythrocytes is RBCs basically. It has your microRNA, mRNA, and all those. You remove this whole thing, you get a sack of bag, and once you have this phospholipid bilayer, they become exactly like liposomes. If you look at liposomes, you assemble liposomes in aqueous, you take these phospholipids at a particular concentration, you create a micellar structure, large micellar structure, lipid bilayered structures. You extrude it through a filter and make it smaller. So it's a very standard technique. Now, will my extruder know that it is a liposome self-assembled or a RBC self-assembled? No, extruder has no clue. So it will not differentiate between them, and it will extrude the the RBCs as well. So that's exactly what we thought. We thought of deriving, nanoparticles from RBCs by this top-down approach. And because it was structurally similar to liposomes, it will have a lipid bilayer and an aqueous core. You can now add drugs in them. And of course, the additional proteins that are present on the surface will give you this extra, the stealth effect, okay? So here is what I was telling you about. You take the RBCs, You break them. Biologists have established this procedure, and you get something called as erythrocyte ghost. They're called ghosts because they're white in color, and they are basically large particles with nothing in them. They also have the capability of sealing themselves back. Okay, the RBCs don't just get broken; they can reseal. So you do the resealing process, and while you're doing the resealing process, you can add whatever drug you require to load, dyes, drugs, whatever. What will happen is during the resealing, they will get inside or they will get arranged. Okay, and then you extrude them. You put it through your filter of whatever size. So if you take micron, like 100 micron or 1 micron, you will get nanoparticles. You can extrude them for a longer time to get until they start flowing. So then they will self-assemble into smaller particles. With this self-assembly, you will have your proteins, your receptors bound, and you will have your hydrophobic drugs inside the core and the inside the hydrophilic core. If it is hydrophobic, then it can be inside the lipid bilayer. You can have dyes which can be inserted anywhere. You can have other polymers as well. So now if you're able to do this, you've opened up an entire process that can be utilized. Okay, so then for our experiments, the drug and the dye that we used was the these are the model drugs, the indocarbocyanin dye and the camptothecin. And what we found was that the reason we used it is because these are hydrophobic. They have very beautiful emission spectrums, so you can actually identify where they are present, whether they are present in the hydrophobic region or hydrophilic region. Camptothecin is a drug which is an potent anti-cancer drug. It's highly insoluble. It's a hydrophobic molecule. In fact, that is the reason why a less potent but more soluble version is clinically approved for lung cancer. So we thought if we are able to develop a system for camptothecin, it can have more clinical promises. So we did that. Okay, we basically loaded these drugs using the same strategy, and we started looking at what have we formed. We started characterizing. So you can see that you have the nanoparticle, you have beautiful bilayer here, the core is empty, you have these vesicles. Okay, we were able to put in drugs and dyes, all of it, because we are able to track them by the fluorescence spectrum. They look bright red here. This is a microscopic image, so they're not aggregating, they are well dispersed. Okay, you can see the size. So this is your RBCs in microns. You have your ghost also in microns, but the nanoparticles were about 200 nanometers in diameter. And once you put the drug and the dye, we thought that it might just rupture the cell assembly process. It doesn't. It actually is in similar range. The zeta potential is basically all negative because remember it's a cell membrane, so it'll be negative in charge. The most important part, after we have confirmed that you can create these nanoparticles, during this process, we could lose our proteins. Remember the CD47? We could lose that. So we thought of doing SDS page and confirming that all the proteins are intact. What is this exact status of CD47? What we did was, what if it is not uniformly present? Some particles have it, some particles don't have it. That could be a trouble. So we started looking at, does the particle size, match with your CD47 presence? So we had this experiment, was very interesting. We had an antibody against it, and we tracked every single particle. As you can see, they all have the CD47 on them. Did some experiments where we started looking at protein adsorption, and they had less protein adsorption, similar to what we have in RBCs. We studied the biocompatibility, and of course, this is more important, you can have less protein adsorption. They were very biocompatible, and you had similar protein adsorption compared to RBCs and the ghost, even after 12 hours, okay? Because it is derived from blood, we thought of looking at the immune response to it, and we found that TNF alpha is a marker for immune response. Whether your immune system has recognized it or not. We found that the immune system does recognize it in the early time points, like 12 hours. However, it subsides down and becomes more self-like after around 48 hours. Okay. On the same thing here, we have IL-6, which is again another marker for looking at inflammation, and we found that that was a little upregulated. So it's not a system which is perfect yet. Okay. What happens to drug loading and release? We found that drug loading and release was present there, but it was very fast release. And we needed to work on that. Anyways, we'll talk about it in some time. So we were able to completely control the drug loading and release. Not control the drug loading and release because it was very fast. Okay. Cellular uptake was all good. They were taken up by the cells quite easily. They were very efficient in vitro. And then we moved to look at their circulation time. Remember, our argument was that if we create this CD47, they will be in circulation for a longer time and they will end up into the tumor for a longer in higher concentrations. Okay, so this is exactly what we looked at. We looked at the organs and we looked at how much is present. You can see that after 24 hours, it was still present in the blood. It started to drop after 24 hours. And in the blood after 48 hours, about 5% was present. About 5% was present. So, most interesting was that this bar, which is showing that these particles can enter the brain, which is interesting because nothing enters the blood-brain barrier. Nothing crosses the blood-brain barrier. So why and how it is happening and how it is cleared at 48 hours is something which we are intrigued by, and we are looking at it. It follows a similar degradation pattern as the RBCs. RBCs get cleared in the liver and the spleen, and you can see that spleen over time had higher concentration, which means that it is being treated by the system as RBCs, and it then sends all the RBCs to the spleen to get degraded, and this is exactly what is happening here. So then we were excited that we have something which is very interesting. What do we do now? So we thought that we'll look at the efficacy in vivo, drug delivery efficacy in vivo, and we'll also look at other applications of it, So as I said, we do not have good control over the drug encapsulation and the drug delivery. We thought of, can we now do a slower release? Because remember, if it gets released in like 2 hours or 4 hours, it's not very good, right? So we need to actually control that release. So what we did was we had a micelle. We thought of making a polymeric micelle in which this drug will be encapsulated, and this will then get encapsulated inside the nanoerythrosomes. Okay, so this is what we did. So we have the erythrocytes, we have the loaded micelles with the drug, and then it will be hemolyzed and extruded, and you end up with something like this. You have a nanoparticle with the micelle. Okay, so now you look at the TEM. It looks a little different. It has the core shell structure, but it's a little darker. Again, did all the characterization, how is the encapsulation happening. We studied all of it in great detail, and it was a lot of optimization that was required. I'm not talking about it. We'll move forward. And of course, the size of the nanoparticle did not change. It was similar, and protein adsorption also was very low. The zeta potential remained, and things like that. It became a little positive or little neutral, you can call it. Okay. So what happens to the, where is it found? As you can see that the tumor, the last one, had the maximum, presence at after 72 hours. So it was in circulation and slowly it ended up into this. So at the end of 72 hours, you can see how it is growing in the spleen. So 6 hours, nothing is there in the spleen. Over time, it increases in the spleen, clearly suggesting spleen and the liver. Liver is maximum in 24 hours, and then it starts to reduce because now everything is gone to the spleen from the liver. Same thing here. So very beautiful. It is working exactly the way we thought it should. And now we wanted to look at the efficacy of this. We tried to do, like combination, like we did CPT. And what we found was that although we were able to reduce the tumor load, so this is how small the tumor became after 21 days, we were not able to eliminate it completely. Okay. So you can see that it is still there and you can see that it is still rising. There could be multiple reasons, but one reason we think is that you can see that after, so blue line, graph A, after like 15 days of staying similar size, after 15 days it increases. Probably because the dosing is very low, and maybe we need to come back and give another dose at 15 days, and then it might remain like this, and the tumor might be lower. Okay. So one way is changing the dosing. So again, because the tumor was there, the survival was also not, longer. After certain period of time, they also died. So it was not the best system. So we thought, is the survival due to just, have we reduced the toxicity? Fine, we have not reduced the tumor load, but what about toxicity? So we analyzed the samples by histochemistry and we tried looking at what is the toxicity over different organs. See, with CPT, which is the free drug, it's a very potent drug. There is metastatic nodules seen in the lungs. Whereas even in the control samples, there was severe lung metastasis, which was observed with this particular tumor model. We did not see that with our systems. Heart, there is, a lot of toxicity associated with the no treatment and the CPT treatment. We did not see that. The liver, there was severe cirrhosis with free CPT, because remember, everything goes to the liver for clearance. And CPT is potent, so it is very toxic. Mild fibrosis, if you do not have any treatment, okay? Same one was with our nanoparticle. So this was very exciting. Kidney, of course, there was severe toxicity for the two cases. Ours was better. The one which was very bad was the intestine. We saw some toxicity in the intestine, which is similar to the free CPT. Whereas in the control samples, there was no apparent degeneration in the intestine, meaning that clearly CPT is being released and causing some sort of toxicity. So we still need to work on that, and we are working on different other aspects, okay, which I'm not going to talk about in today's talk. Moving forward, so clearly, I'm sure by this time I've convinced you that changing the, so I think I have another, an hour, right? Or maybe 45 minutes. So I'll try to wind up everything in next 30 minutes. So otherwise it'll become a very long talk. Okay. So clearly we were able to, now you see how beautifully we were able to design a system from nature, understanding these things. And we thought that, this is something different. We started looking at more aspects. For example, what is the surface lipocity, lipophilicity, and what happens to the uptake? How does the nanoparticle, so the question is, how does the nanoparticle surface affects the uptake? So why did I ask this question? Because remember, the cell membrane has a lot of proteins. Those proteins can be hydrophilic. There could be hydrophobic proteins also. If you look at the charge, there is the charge distribution, some might be positively charged, some might be negative. What you get from a zeta potential is an average, right? But the region, if you want to look at the spatially, what is the region like, they will be different. There will be, a patchiness in the surface, okay? So we thought, can we utilize this patchiness in the surface to do something? This also, at the same time, we also looked at one paper which was talking about elasticity of the material. So for example, if you have a very rigid material, it enters the tumor more, whereas a softer material does not, and things like that. Okay, sorry, exactly opposite. A rigid material, this is megapascal, and this is kilopascal. Okay, so this is basically a beautiful study which was reported in Nature Communication a few years ago by Deborah's group at Harvard, and they showed that there is a difference in the uptake based on the nanoparticle that you use. The argument they gave was, by the way, there was an optimum. It's not like very rigid, less rigid. There was an optimum rigidity which was required. Okay. So the argument they gave was, if you have this liposomal structure, it can actually deform, okay, and it can enter the cells better if it is less rigid. Whereas the other argument is that it can also fuse with the, nanopart with the cell membrane, and it can enter. Okay, so there are multiple pathways: fusion, endocytosis. Hard particles enter via certain methods, soft particle enters via certain methods. Okay, so now the question is, fine, it enters here. Now if I have to utilize, I'm going to skip this RNA thing, so, or maybe I will talk about it. So suppose I want to utilize, so they enter the cells. Okay, so we are here, we are inside the cell now. So the first step was, we have tackled the extracellular interaction. Extracellular was your protein adsorption. So we have tackled that. Now the cell is able to enter. I have this beautiful RBC particles which are taken up by the cells. Once it enters the cells, this needs to interact with the cell organelle. An example for that is RNAi. And again, RNA interference is a very common technique used for biomedical engineering, problems, a lot of problems associated with various diseases. Quickly telling you the mechanism. What happens is that you send the nanoparticle, and there is something called as the RISC complex. Okay, so RISC complex is, you have your protein. It's a protein. It's a big protein. It's about 30 nanometers in size. Okay, and you have your nanoparticle with your duplex of the genetic in materials. For example, siRNA or short interfering RNA. So you could have DNA or RNA. So here it's an RNA. Usually it's a duplex because duplex are more stable. A single strand of RNA is very unstable. It will get cleaved by the RNases, whereas a duplex is a slightly more stable. So you have your duplex, and you can see here, this duplex goes there, and there is a processing that happens. Basically, the passenger strand is removed, the the coding strand is taken up and understood, and then basically it interacts with the RISC. Let's not get into biology too much. And this is the translation process, which you all, those who have studied biology, would have studied it. So let's not get into the biology too much. Let's do a simple thing. You have a nanoparticle. Maybe I have it over here. Yeah, so here. So you have a nanoparticle, you have your siRNA on it. First of all, it's double-stranded, so it has to be unwinded. But let's say it gets unwinded. Then you have this particular protein. This section of protein is called as the AGO2 protein. This protein needs to sit on this small section. Let's get you the scales. This is 30 nanometers. This is 30 nanometers. This is 20 nucleotides, few angstroms. Right? Very small. Think about it, a big ball with a small strand lying, and this ball needs to look at that strand. Will it be able to do that? No, there is a lot of sterics, right? So it should actually not work. So RNAi with nanoparticles should not work, but it's still working. So what can I do? If it is a hard material, this steric will be there. If I want to reduce the steric, I can change the elasticity. Suppose I make it elongated, and it elongates, and it can now interact with the siRNA easily. So here, is the argument. I can either make it smaller than the nanoparticle, I can make it, I can have like a, I can change the elasticity of the nanoparticle, right? And then I can study what the data, what happens, because these kind of studies are very important if you want to make better vehicles, right? So we started looking at this in the lab, and we started looking at whether, you have so we had 4 different systems: a small particle which is soft, a large particle which is soft, a small particle which is hard, a large particle which is hard. We characterize the nanoparticles, and remember, the surface has to be similar charge and similar size and all that. Okay, so I'm going to skip the characterization. They were biocompatible, their stability and everything was there. They were taken up by the cells very easily. The most important part, and we were able to load siRNA on them. Okay, and we were also able to release the siRNA. So here it is. Here is the interaction. You have your nanoparticle, you have your siRNA, you have your AGO2. This data just suggests that the nanoparticle was conjugated. So you can see that siRNA, this is the siRNA band, and the green, is the nanoparticle. So this is the nanoparticle, or the protein, sorry, the protein. This is the siRNA, and they were able to move and interact. So whether the interaction is happening or not, we will look read out by this interaction with this. Okay, so here the nanoparticles were basically interacting with this protein, and we saw that we have to study this interaction. So this is just a readout. So here is the experiment we did. We had the siRNA, we had the AGO2 protein. If it interacts, there is a dye called ETBR that gets intercalated, and if it intercalates, you basically get some fluorescence, and that fluorescence would be a readout. Anyways, by doing these experiments, the 2 assays, that in-house assays that we developed, the ethidium bromide assay and the AGO2 interaction assay, we confirmed that a small soft particle is best for this interaction, followed by a large soft particle. Remember my argument where I said that either it has to be small, so that I understand, or it has to be, so soft also I understand. So if it is large, it has to be soft. What was surprising was this one, that a large hard particle is better than a small hard particle. And we were very surprised by this result. We were like, what is happening? Well, now if you look at so again, we confirm that this is the real effect, and we are actually seeing that we are looking at gene knockdowns. So this is the mechanism that we developed. What is happening in the soft nanoparticle is, you have your protein. It is easily interacting. It's soft, so you have a deformation of the nanoparticle and nice interaction happens. If you have large particle, if it is soft, it can interact well. If it is hard, how is it interacting? It has more surfaces for it to interact. It actually looks at it as a local region, which we missed out when we were looking at single particle interaction. Okay, so you have large surface for interaction. Okay, whereas if you have a hard particle, so if you go here, this was worse because it is small in size. There are not enough samples where it can interact, and it is really, really hard. It cannot deform. So the worst is hard nanoparticle, which is small in size. This information actually, with our hypothesis, we could have never obtained, okay? Because we never understood that it is making almost like a flower structure. So we did cryo also. I'm skipping all that data here, but we did a lot of confirmation of this aspect. Okay, anyways, so again, we had the first few minutes that we talked about, we had a nanoparticle where we used RBCs. We removed the protein interaction. They were able to reach the cells. Once they entered the cells, now they'll be able to interact with the proteins better, right? So we have kind of understood all these processes. The next is, Fine, we've been able to develop these beautiful systems. What happens to Ross generation? The Ross will still be generated, no, because these are nanoparticles and they can be catalytic. So we started looking at, and as I said, Ross generation leads to a lot of DNA damage and other things, right? So we started looking at some strategy of reducing Ross. So, and Ross generation is a bigger problem with graphene or inorganic materials, smaller inorganic materials. In fact, we were quite shocked to see a lot of literature in 2011 and 2010, which talked about nanoparticles which are biocompatible. So here, you see that strong green fluorescent particles for bioimaging. And if you look at the toxicity, just by 400 microns, 20% cells were dead, and they're calling it biocompatible. Same thing here. If you look at this blue graph, about 1 milligram, 50% cell death. So these are not biocompatible. Why is that happening? Again, I told you there could be Ross generation, and the toxicity is probably because of the Ross and because of the small size. Because remember, for quantum dots, you want them to be smaller, because otherwise you make them bigger, you lose the quantum effect, you lose the fluorescent property effect of that particle. Okay, so you have your Ross generation, you have your oxidative stress which causes and things like that. So I'm going to skip all this. Then people started looking at in 2012 there was a report talking about how size affects it, and of course, indeed, this argument of size, which interacts with the protein folding that I gave, was proved by this. Okay, so that's where we understood that it works like that. So now the question is, we want a carbon dot or a quantum dot based system. You want to keep the size small, but you want to create some masking of the Ross. So the Ross should not get catalyzed on the surface. So very simple, we thought of putting a polymer coating on it. Remember, we are not the first one to put the polymer coating. Many people have thought of it. They have done it. However, if you have a very thin layer of polymer coating, it is not enough for the Ross to get quenched. Okay. So we thought, can we make a bag and put our nanoparticles inside them so that there is enough polymer, the nanoparticles are dispersed freely, they still maintain their quantum behavior. Okay, so that's exactly what we did. Again, I'm going to skip the details of it. You can read our paper if you're interested. Basically, this is what they look like. This is very, like a elongated structure with a lot of nanoparticles embedded inside them. The photophysical properties were retained, so they were fluorescent. So as I said, we focused on looking at the Ross generating ability. So you can see that with your, this is the PGQD is the polymer coated. If you do not have polymer coating, the Ross is increasing after 90 minutes. Whereas if you have a polymer coating, see how dramatically low this Ross generation is. We were very excited and we started looking at whether this will be replicated. So this was done in a tube, okay, in a test tube. So will it be replicated with H2O2 as the Ross generator or the free radical generator? Can we see the same thing in vitro in cells? We actually did imaging of these cells and started looking at what is the Ross generation. So Ross generation means it is going to kill the cells. Here you can see that the nanoparticle is blue in color, so the cells become bright blue. They are entering inside and the cells are very healthy. You can see the health of the cells. They are beautiful, healthy cells, okay? So we have intracellular Ross also here. And then, you can see that inside this, the bright green color means very high Ross quantity. Less green means low Ross quantity. So we had less Ross present by these things, intracellular Ross, which suggests that they are not toxic. Again, we started looking at, does it really reduce the toxicity? Can it actually deliver drugs? And we did all these experiments because now we have the gels, we can actually load it for drug delivery also. And that's exactly what we found. We were able to have beautiful nanoparticle which are coated with these polymers, and they were able to deliver drugs in them. They had less toxicity, and we were able to improve the toxicity. So for the model system that we used for our study was graphene quantum dots, because graphene quantum dots have a lot of severe toxicity associated with them. So here you can see that we were able to we were able to reduce the toxicity to about, so 5.5 milligrams showed almost 70% cell viability. So only 30% cell death, whereas compared to other results, that I showed you earlier, where you had less than 1 milligram and showing 50% cell death. So clearly, and here you can see here, with 5.5 milligrams, it's extremely toxic. But with PEG, the toxicity is reduced. So we've mitigated the toxicity of the drug, okay? With that, I would like to end the talk. And, I hope that, I've been able to convince you how we have developed our nanoparticle systems for drug delivery. These are all advanced systems by fundamental understanding, by understanding what kind of cellular interactions happen and trying to address those interactions, okay? So, all this work would have not been possible without the funding agencies. So I would like to acknowledge the funding agencies. I was very lucky to get an initial funding from the DBT through the IYBA scheme. DST Nano Mission has funded our elasticity work. DBT Nanobiotechnology has funded our RBC work. IYBA has funded the carbon dot work. And now we are funded by the Janki Amal and Sir Power Fellows is funding our work. And apart from funding agencies, money is important to do research, but the main source which is doing this is my manpower, and I'm very lucky to have really talented students in my lab. So all this people work really hard in my lab, and they are the ones who actually do the science. So it's been a fun journey with them. And I would like to thank you all for patiently listening to our work and giving me this opportunity. So I can answer any questions now.
Okay, session is open for question answer. Participant, you can ask any question, or you can type in a chat box.
Yeah, I'm looking at the chat box. So okay, thank you. I'm glad that you all are liking it. It was a long talk, but I think that's how the session is. So. No questions? Okay. In case you have any queries related to, drug delivery or what I talked about, I know it's a lot of information to, process. Okay, there's one question. Can we use same or similar nanoparticle for drug? Yeah. So now we are exploring that. In fact, the next work with the RBCs is we are exploring, dual delivery of, chemo as well as photothermal therapy. So we are looking at the synergistic effect of these two, and we've seen some very promising results. So yeah. See, the best part of these RBC particles is that there are challenges for clinical translation. For example, it has to be patient specific and things like that. But if we take blood from the patient and we have some process of developing particle then and there and giving the injections, then you get rid of all the toxicity that is associated. So we are trying to look into those aspects. The next few years will be important for that. Okay, so what is the difference between in vivo and in vitro? Okay, so in vitro, when we do it, so there's also a term called in silico. So in silico is computational work. In vitro is basically everything done in a petri dish, So you have cells and other things, you stimulate the environment which is there, and you use that conditions to do the experiments. So that is in vitro. In vivo is in animals or in host, in a real living organisms. Okay. Is nanoparticle toxic for human being, and how we can remove from body? It's a good question. The toxicity is there. So I just talked about the reasons for toxicity in today's talk. How do you remove it from the body? So one is to degrade them. So they will get degraded and cleared from the kidney. And other one is through, they might, so for example, the RBCs, they get into the spleen and they get degraded over there. So, they will eventually be cleared. Simple. Inorganic material is difficult to remove. For example, gold nanoparticles, very difficult to remove, but then they're not toxic. They remain in the liver for a long time. They don't cause toxicity. High concentration can cause fibrosis and other effects. So that is a challenge. Yeah, Dr. Jairam,
Yes, madam.
I think there are no more questions.
Yes, madam. There is no more question now.
Okay. So shall we end the session, or what is?
Wait, ma'am. Janvi.
Yes, sir. It's a great pleasure to propose vote of thanks for this session. I thank today's eminent speaker, Dr. Neetu Singh, for her valuable time and deep insights shared by her on the smart functional nanomaterial that is drug delivery, biosensors, and biomedical implant coating. She covered integrating concepts and skills from material science and biology to design nanostructured material with the enhanced functionality for applications in the biomedical implants and drug delivery. She particularly focused on seeking the systematic understanding of the bioactivity of the nanostructures. We are grateful to you, ma'am, for the knowledge you have shared with us, and I'm sure that all the participants would get benefited from it. I thank all the participants from their enthusiasm and active participation. Thank you so much. Hope to see you all for the next session. The next session will be at 3:30 p.m. on the same link.
Thank you so much, Dr. Gulave, for inviting me. And it was a pleasure, as I told you earlier. And all of you, in case you need any help, any questions, anything, feel free to send me an email. And, I would love to interact with you in person when I come to Mumbai next time.
Okay, thank you. Thank you, madam.
Thank you. Bye-bye.
Bye. Yes, it's very fine.